Hello everyone, my name is Desmond Singh, and welcome to my poster presentation titled A Weighted Resampling Approach to Differential Expression Analysis. So, differential expression analysis, or DEA, is a statistical method that allows researchers to provide quantitative support for changes in gene expression. While originally developed for analyzing microarray data, it's now more commonly applied to high-throughput RNA-seq count data. Most popular DEA methods use parametric statistical models, but can face issues such as poor fit by the assumed distribution, usually a negative binomial distribution, no gene-specific variant stabilization, and ineffective FDR control. The McConkey lab at UWaterloo has developed a potential solution to these issues, RAID. RAID, or a weighted resampling approach to differential expression, is an R package that implements a non-parametric algorithm for DEA. More specifically, it combines observed gene data with informative priors to stabilize the variance of individual genes and uses resampling to compare treatments and assess statistical significance. Now, given an RNA-seq count dataset with J genes, N samples, and at least two groups to compare, RAID performs DEA in three simple steps. Let's explore these steps by observing how RAID analyzes data for two genes within the TCGA bracket dataset. The first gene, PUM1, is stably expressed between tumors and normal tissues across different cancers. Here, it's considered a negative test case, as we expect RAID to report little to no differential expression between tissue types in the TCGA bracket data. The second gene, MKI67, is upregulated in tumors across cancers. Here, it's considered a positive test case, and we expect RAID to report its significant overexpression in tumors within the TCGA bracket data. And when we actually visualize the TCGA bracket data for both genes in 6 participants per tissue type, we can broadly see that our data align with our hypotheses. So, the first step in RAID's workflow is to construct a prior reference pool. This is necessary, as the reference pool identifies genes whose data will be used for variant stabilization. A pool is determined for a given gene within the dataset by identifying a window of 100 genes within the dataset that have a similar mean CPM expression across all samples. Then, of those 100 genes, RAID identifies which ones have coefficients of variation that fall into the central 80% of their distribution. The full reference pool for the TCGA bracket data across all genes is depicted here in Figure 2. Points colored red reflect genes whose individual measurements will be used during variant stabilization. Once a reference pool has been resolved, RAID proceeds to its second step, which is generating cumulative distribution functions, or CDFs. Now, generally speaking, the reference pool will yield a prior CDF for each gene by condition, for example, PUM1 in tumorous tissue. Experimental data for each gene by condition are organized into an observed CDF. Corresponding prior and observed CDFs are then combined to yield an output CDF, which will have its variance stabilized and be used for downstream DEA. The complete set of CDFs for both PUM1 and MKI67 is depicted here in Figure 3. For the former gene, one can observe that the range of expression values is similar across both tissue types. For the latter gene, there's a much broader range in tumors versus normal tissue. RAID then uses the output CDFs for a given gene across experimental conditions to generate a resample distribution of 100 log2 fold change estimates. The details of the resampling procedure are outlined in this text box, and the resample distributions for both PUM1 and MKI67 are depicted here in Figure 4. We can see that the distribution for PUM1 is centered at a log2 fold change of negative 0.31 and contains a dashed black reference line in its mass. This line refers to a log2 fold change of 0, which indicates no differential expression of PUM1 between tissue types. The distribution for MKI67 is centered at 2.84 and does not contain 0 in its central mass, suggesting marked upregulation of this gene between tissue types. Now, having generated log2 fold change distributions for each gene, they are then used to derive p-values, which estimate the probability of the distribution containing a value of 0. The set of raw p-values and log2 fold change estimates can be visualized on a volcano plot with a raw threshold to assess significance. Figure 5 depicts such a plot with both PUM1 and MKI67's data points labeled in red. One can observe that, as expected, PUM1 does not appear to be significantly differentially expressed between tissue types, while MKI67 appears to be strongly overexpressed in tumors. Now, based off of this preliminary work, one can conclude that the reference pool provides an effective distribution-free method of variance stabilization, the use of prior CDFs enables resampling with low replication, the significance of non-zero effect sizes can be readily incorporated into this method, that RAID was effective at identifying differentially expressed genes without an assumed distribution, and that resampling is a valid alternative to parametric DEA. Very quickly, I wanted to say that this work would not be possible without the gracious funding provided by NSERC as both a grant and a scholarship, and I wanted to thank you, the viewer or listener, for viewing my poster and letting me talk about my work. I hope you enjoyed the conference. Take care.